So I'm going to talk about the SMC challenges, and in particular, I'll talk about the status of SMC-Het and SMC-RNA, and this is one of those fun talks where you get to talk about things that didn't work well and things that did work well. So the origin of the SMC challenges, or the ICGC-TCGA Dream Somatic Mutation Calling Challenges, is the idea that if you're doing a cancer analysis, you will often have complex information on a large number of patients. And as we were just talking about, you need to do some sort of pre-processing, which is a little bit of a black box. And how you do that pre-processing will hopefully lead you to some exciting endpoint. And by doing that pre-processing, we are locking in some understanding of how the data set fits, what are its noise characteristics. And it's well understood that how we do that pre-processing leads to, unfortunately, different results in our end conclusions and our understanding of the biology and the predictors that we might construct. And so you can find numerous examples of this in the literature. I'll give you one. This is a study looking at predictors of outcome, relapse or overall survival, for non-small cell lung cancer. And there was a couple of biomarkers that had been looked at, and there was an assessment of how you might find variability in the predictions of those biomarkers if one took a look at multiple ways of pre-processing the data. In this case, it's microarray data, and we're just talking about different normalization techniques. And the answers are striking. Only 150 of 450 patients, so about a third, showed the same predictions of a biomarker when one changed the way that the data was normalized. And some of these are not surprising. Sometimes normalization can be quite noisy, but some of these are really just different parameterizations of the same model. And even that is enough to change the way that a patient's outcome might be predicted. And that's true for all tumor types. Here we're looking at a set of 48 normalization methods as the columns, and the rows are genes. We're talking about breast cancer, and each of those black squares is whether or not an individual gene is predictive of patient outcome in about 2,000 breast cancer patients. And the numbers are scary because 3/4 of genes are associated with outcome by at least one normalization method. So if you tell me, Paul, I really want to show that gene A is associated with patient outcome, then I just have to look across 48 normalization methods and I've got a 3/4 chance of finding your result for you. The problem is less than 16% of genes were in a third of methods, and not a single gene was associated with outcome in all of the methods assessed. And of course, this holds in now new next-generation sequencing techniques. On the left is work from the TCGA Singer-Maas group looking at the renal cell project, 300 patients, and it's the calls of point mutations from the exomes from Baylor, from the Broad, and from UC Santa Cruz. And you can see that there's a nice core of things that everybody agrees on, 12,000, but there are thousands of discrepant calls from these groups, major international sequencing centers. Similarly, on the right is work from the ICGC in our group looking at different ways of identifying somatic structural variants, and the overlap is not good. And so to first try to resolve this, we started in 2013 launching the SMC challenges to do global standard benchmarking. And the first challenge took about a year, taking a look at multiple data sets. At this point in time, we redistributed sequences by shipping them across the internet, and we had both simulated and real sequences. And this was very successful. It drove changes in the pipelines that people were using at many centers, including in the ICGC and TCGA themselves. So there was a lot of ability to be able to take these data and drive them to both interesting conclusions for algorithm developers and a better insight for how one might standardize pipelines. One of the most striking results that came out of that was the association of error profiles with the cellularity and complexity of a tumor. Here we're looking at just somatic single nucleotide variants, so a single point change in the genome. And on the left is a tumor which is 100% cellular and it's monoclonal. It doesn't have any subclonal complexity. And on the right is a tumor which is somewhat contaminated with normal stromal cells and has 2 subclones in it. And if you'll notice, on the left, you see groups that get to almost 98% sensitivity, 98% specificity, very, very close. On the right, now there's almost like a a hard wall where no group is able to exceed a recall of 90%. Essentially, the combination of the modest coverage sequencing, this is about 80x, and the subclonal complexity makes it very difficult to be able to accurately identify all of the mutations. And so that led us to the SMC-Het challenge, which was an attempt to try to understand and articulate subclonal complexity. And that title is well chosen because the SMC-Het challenge has been challenging. The idea was that we would take whole genome sequences, and from those whole genome sequences, we would get contestants to ask and answer 3 basic types of questions. What is the composition of the tumor? To what extent is it pure versus contaminated with normal? How many subpopulations are present in it? What are their cellular prevalence? How many mutations are there? And then to ask questions about, okay, well, we know how many subpopulations are there. What characterizes them? What mutations go uniquely into each of those? And then finally, once you know those subpopulations, how do they relate to one another? What's their evolutionary trajectory, and how do they delineate the complex history or evolutionary archaeology of that tumor? The challenge was designed around the idea that you couldn't come up with a ground truth for these type of questions without really deep single-cell sequencing, which didn't exist at the time. So we used the simulator that we had developed in SMC-DNA to simulate a series of 50 tumors with very realistic characteristics that we'd seen from real tumor sequencing data, as well as some special cases to test key hypotheses. And then we had participants giving us Docker containers, and they would be able to use those and execute them on germline and somatic information and use those to be able to make predictions. That was the idea. There was a lot of things that had to go into developing that. The simulator needed dramatic expansions because it didn't account for a lot of the signals that are found in subclonal reconstruction algorithms. Those include phasing of mutations across the genome, allowing for whole genome duplication, replication timing, etc. In addition, to our surprise, there had not been methods for scoring subclonal reconstruction before. In fact, even the phylogenetic literature didn't have a lot on how one might quantitatively assess and compare methods. So there's a lot of new things that had to be done to develop this. And everybody in the room knows where our problems come because everything that we developed new ended up having challenges. Since last year's DREAM meeting when we were writing the paper, we found a major bug in the simulator where our random number generator seed was not properly set, and subclonal variants were bashing on top of clonal variants, making the complexity of the tumors dramatically less than expected. There was also a bug in the scoring harness which led to numerical failure for a subset of cases, a small subset of cases, but unfortunately the exact subset that we had not scored prior to last year's DREAM meeting. So aside from fixing those, we have now spent almost 20,000 CPU hours redoing all of the computational aspects of the challenge. That is frustrating. But finally, we do actually have results, which is exciting, and the results become very clear. And I'm just showing 1 or 2 key points in this complicated figure here. We're taking a look at a series of tumors, the columns. We're taking a look at just 2 of the algorithms that are used for scoring. And we're looking at those tumors across different coverage depths, and we can see the dramatic increase in accuracy as a function of coverage. We can also see the strong sensitivity of subclonal reconstruction to the algorithm that's used to be able to do the variant calls up front. And we can show that the perfect algorithms do much, much better at being able to make those predictions. So we actually have happy results and are now deeply engaged in writing manuscripts. There's still 2 planned from this challenge, a design and tools outlining all of the work that I described in simulator and scoring, and that's in the late stages of drafting, and we hope we can get that out to Nature Biotech this year, and then the challenge results paper, which will be early next year. One of the lessons for DREAM is that we did dry runs, we did multiple training rounds, and we still didn't identify some rare but very, very important bugs that made our life difficult. So after we finished the difficulties of that, we launched something that we thought would be easier, which was an RNA-seq challenge. And happily, easier turned out to be easier and to be successful. The idea was to have a benchmarking effort to improve methods for identifying cancer-associated RNA sequencing aberrations. That meant isoforms that were tumor-specific and be able to detect and quantify them, and to be able to identify fusions and be able to identify not only the fusion is present, but also its specific breakpoint and isoform. Here we used a fairly complex execution mechanism with Docker containers again being submitted. We ran those Docker containers on the NCI Cloud pilots, and much like the DM challenge, contestants never see the data until after the challenge is fully completed, and this has the advantage of creating a library of identically formatted tools. That was a big advantage in this space, where it turned out that the precise input and output definitions of pretty much every RNA-seq tool varied a little bit, and this brought them all into a common format. So our submission structure really took advantage of the NCI Cloud pilots. We in particular used the ISB and 7 Bridges clouds as the two most heavily used, but all 3. And those were critical to being able to do the large amount of scoring that we needed. The challenge closed a few weeks ago. We had on the order of 50 participants from about 20 teams. Scoring is in progress, and we've released the simulation pipeline, and so far things look happy. We have already some really surprising and interesting results, one of which is shown here. This is a set of teams along the columns, and the y-axis is the F1 score of predicting fusions. And you'll notice that most but not all teams do dramatically better, dramatically 10 to 15% higher F1s in predicting interchromosomal fusions rather than intrachromosomal fusions. And this is a precision sensitivity plot of those predictions, and that is a really great scatter plot of some teams doing really, really well and other teams doing very poorly. And in fact, there are multiple tumors here, so there's incredible intertumoral variability in quality of predictions. So I'll close by just thanking the people who ran these challenges with me, in particular Josh Stewart and Kyle Elrott from UCSC and OHSU, who are critical in running both of the 2 challenges. Paul Spellman's group, who's been doing the real data generation for SMC-RNA, and Peter Van Loo, David Wedge, and Quaid Morris on the subclonal reconstruction challenge. Thank you, and I'd be happy to take questions.